So we have completed the portion of regarding cell, and now we are entering into the next more deeper section, that is genetics. So far we know many things about cell division and replication, etc. We have seen that what importance chromosomes are having. So you know much about gene, so I'll not go for details about gene, what gene is, and how it is there. I'll expect that you go through this. Anyway, I have asked you to read the topics earlier, so I'm sure you might have read it. If not, please go through these topics. This will help you. So you go through the genes, what the genes are, and what is their importance. So we know that what's your inheritance is there, what's your information about the structure and function of any organization, which it carries from the previous generation to the next generation. This is all present in a form of thing which we call as a gene. So they contains all the information necessary for the living cell to survive and reproduce. And these are made up of DNA, we know about it, and the sequence is very important about that particular sequence that is responsible for certain meaning of it. You call them as a codons, and these codons are either carried as such as in the daughter cells, or these codons are used, or this sequence is used to make this copy of it in a different way, that is called as a transcription, and DNA is copied as a RNA, or DNA is having another set, that is RNA, and this DNA to RNA is there, which can be either non-coding. The RNA can be non-coding RNA. It may have a direct function, this non-coding RNA, or an intermediate messenger RNA, and that will be then translated to proteins. So this is how the gene is working. We have discussed so many times why we are studying this, because basically or ultimately the cell function is depending on that genetic structure, what kind of structure they are having, and this genetic structure will determine the structure and the function of cell. So anything goes wrong with this structure and function, it is because of these genes. Okay, because we know that all cells, whatsoever functions they are having, it is because of again their structural or functional things which are present. Now it can be what? This can be structural proteins, or this carbohydrates or lipids. These are the basic blocks. So these basic blocks, they are having certain kind of structure, and because of that they are forming a typical structure, and because of that typical structure they can perform even functional activities also. So we know about it, what are the structural and functional. I told you, you write down all the structure and functions, or all the structural aspects. Receptor is a structural aspect, cell membrane is a structural aspect, okay, size, shape is a structural aspect, and on that basis we have so many functions. Receptor, it has got a function. In receptor, after receptor there are so many enzymes, so many chemicals in the form of maybe hormones, in the form of lipids, in the form of proteins, amino acids the body is producing, and for all there is a requirement of certain kind of enzymes. So that's these proteins, and these are important, what we can say, catalytic moieties, which are taking place or which are playing important role. So therefore genetic structure is important, therefore this genetic structure, how it helps, is important. We know that sequence are there, they are causing encoding, so we call them as a codons, and these codons are giving you about type of protein, how amino acid sequence will be there, and thus this sequence will be important in the structural and functional aspect of any kind of biological molecules. Most of the genetic structure is broadly similar in eukaryotes and prokaryotes. They too have DNA, we too have DNA. In DNA, riboxylic acid is there. In RNA, only riboxylic acid is there. Okay, So these are there. Then many things are there. Nucleotides are mostly common. So these are basically most of the things are similarly. Only slight changes are there because of their type of processing, or we call it as gene expression. So there are things are there. So there are basically three important things, that is gene annotation, gene expression, and gene functions. So how the gene is present, how gene is expressed, means how the gene is either replicated or transcript, and what are the functions. So these are the basic things which are there. Uh, so it is a process. Annotation is a process of identification of location of genes and all the coding regions in the genome, and determine what this gene will be do. So it is sort of a note which is added by way of explanation or comments. If you see PowerPoint presentation, you will find annotations behind below that. Okay, so there are such kind of annotations are there. Then expression of it, how it is expressed, by which information from gene is used for functional product of gene, and lastly exact function what it will be. Okay, we call it as a phenotype, that is because of that particular genotype what kind of function will be there, so we call it as a phenotype. So basically three things are important with this gene. Now when we see about it, what are the common features, again you have studied both ribox sugar is there, or deoxyribose is there, and what happens because of that, you know about 3 5 or 5 3 everything is known to you already. We have discussed it. I request you please go through it again, so that we can save time, because we will not repeat into it. Then nextly, the regulatory sequences which are located at the extremities of the genes, and they are causing for various kind of activities. How many portion, what type of portion is actually should be done. We have got various kind of agent or what we can say moieties, or various kind of molecules. They are acting as a promoter gene, which causes the increase in, or which causes initiation of the gene expression. There are certain genes are there, or certain areas are there, certain chemicals are there, we call them as enhancers of that. So they enhances or increases the gene expression, and there are certain silencers which will stop, which will be decrease, or which will keep it in a nascent condition. So these are variety of chemicals are there. We shall be seeing them one after another in due course of time, or when we will go for various topic in this. So the proximal promoter region that will bind to the transcription factor, again there are variety of transcription factor, and they are responsible for again variety of activities, and this will modify the affinity of core promoter for the RNA polymerase. We know about that when transcription takes place, when replication was there, we call it as a DNA dependent DNA polymerases or DNA polymerases. Here we call it as a DNA dependent because it is depending on a DNA. So we nowadays we only call it as a DNA polymerases or RNA polymerases. So they will give rise to sequence of the RNAs, and therefore we call it as RNA polymerases. Genes may be regulated by multiple enhancers or silencer sequences, which can further modify the activity of promoters by binding with the activator or repressor proteins. So these are again variety of activator and repressor proteins which are participating in the gene expression activity, or what we can call it as a transcription activity. These enhancer silencer may be distantly located from the gene, or may thousands of base pairs away, but whenever the time will come, they will accordingly do their job. Binding of different transcription factor therefore regulate the rate of transcription initiation at different times and in different cells. So therefore this variety, why we can able to produce variety of action. At some time it is too much, at some time it is not less, sometime it is less, sometime it is different, sometime only few things are synthesized. So if you think silently over it, then you will find that how complicated this system can be. And still this system is working, still it is able to either increase or decrease the activity. It can increase or decrease some of the molecules synthesis, it can increase or it can stop at some time. Okay, so there are so many activities are taking place. So how this system is working, so many chemicals So many proteins, enzymes, molecules, biological ligands are working into it, and they are responsible for this very complex regulation of this. So regulatory elements, they can overlap one another in a section of DNA to interact with many competing activators and repressors, and some repressors protein can bind to the core promoter, and they will prevent the polymerase binding. So like therefore it is repressor means which will inhibit or which will stop. The genes with the multiple regulatory sequence, the rate of transcription is the product of all elements which are combined. Binding of activators and repressor to multiple regulatory sequence has a cooperative effect in a transcription. Again, certain areas, certain portion of the genes may be responsible for multiple products. Now, because of the presence of these regulatory things like repressor or enhancers, they will make what? They will cause enhancement of certain one and decrease in certain type of chemicals. So same thing, same genetic makeup can able to make the difference in the activities. Okay, so these are the reasons, these are the what we can say explanations for this. Now we can see the how this system is working. If you see this figure, so this is the regulatory sequence of DNA. So this is a DNA molecule. Okay, so the silencer or enhancer, what any kind of that particular molecule will come, that will bind, that will then cause initiation or unwinding of DNA. We have seen that DNA has been coiled or supercoiled, then it will start it. If it is enhancer, if it is silencer, then it will be stopping it. It will cause it to get it wound again. Then there will be promoter area will be there. It is a sequence of a DNA where the proteins bind, and they will initiate the transcription of single RNA from the DNA downstream of it. The RNA may encode a protein. Again, this protein which is there can have a function, and or itself it can do job as a transfer RNA, messenger RNA, or this kind of activity. So then next is the 5' untranslated region will be there in that. This region which will there is not is present directly upstream from the initiation of the codon. We call it as a promoter. Where you're binding, we call it as a initiation codon or starting codon many times. This region is important for the regulation of translation of transcript by a differing mechanism in viruses, in prokaryotes, in eukaryotes, and these are untranslated. This portion sometime can take part in the translational activity. Then start codons will be there. This will be causing the actual start of the transcription, and then accordingly from this you will find that RNA is formed from that. It will cause opening of the reading frame, that is whatsoever area which has to be read from where, from the DNA, this area has to be read, and accordingly this area will be read, and then lastly there will be stop codon. Only that much area between start and stop codon will be read, and that much copy of that will be in the form of this RNA will be there. Okay, so this will be the last portion, that is 3' UTR, and again the next sequence will be there. Either there will be enhancer or silencer molecule will be there. So again it will start from that, how it will be there. So here it will be termination of that much area will be terminated. So this is how this happens. We shall be studying in one by one steps, in one after another one. So this is how genetic process is there. Now again you might have read it into it, when transcription takes place, then this is a whole sequence. Out of that there are certain areas which are functional and other areas are not functional. So we can see that these are the exons, and there are introns which are not functioning. Okay, so this much exon, this much exon, this much exon, and these are the introns. So this intron has to be cut down. We call it as a splicing of RNA. If you have read these terms earlier during physiology, then it will help you. So and only exons are there. They will be combined, and introns will be removed during the process of splicing. Now this will be there, and that will go for protein, this will call as a protein coding region, that will go for the ribosomes, bind with the ribosomes, and this area, because of this, the protein will be formed, which will be functional protein. So this is how this gene and gene function takes place. We shall see again the details whatsoever are there, how they are taking place, and what are the various aspects of this. Now again, when many times jokingly I used to tell you, whether fortunately or unfortunately, why I am calling that is, unfortunately the details are too much, too many details are added, so you have to study many things. However, fortunately means because of the understanding of this all information, maybe we have to study it more, but because of the understanding, many new drugs, many new pathways, many new mechanisms for understanding the basics of the pathophysiology and then possible treatment for that, these all areas are opening, and because of that many new treatments can be possible. So that way it is fortunate, because it is helping mankind because of this particular thing. So these are various ways how it is taking place in eukaryotes and prokaryotes. No big difference is there, very small differences are there. You have already studied it, you know about it. So please go through this and how this works. So this is what we call it as a genes. So what are the genes? We know genes are again what kind of structure it is having. Basically it is present, it is if we'll go for the chromosomal structure, it is a spiral one. The DNA is in a spiral form, it is bound, there are two strands, everything you know about it. Okay, and this we call it as a Watson and Crick model. So that shows how it is, and this will inform you that this various type of nucleotides and are present, purine and pyrimidine types are present, and they are arranged in a particular sequence. This sequence is very, very important to what you called as a codons. These codons are playing important role in variety of activities. So this all thing we have studied, you have known about it, so please go through it. Then next thing is how the this is elucidated. Okay, so how, because earlier it was not knowing, nobody was knowing, and it was on the basis of trial and error basis it was happening. Okay. So this latter, what happened is, fortunately so many instruments, so many equipments are were there, and because of these equipments one can go for variety of actions, variety of things which can be done. So this is what called as a molecular genetics, and this molecular genetics is possible because of many things. So simultaneously what happened during the period that there is a technological improvement also, that is we call it as the biophysics. So because of this biophysics knowledge, it has increased. There are so many techniques are available to check it, to confirm it, and because of this variety of things, now as I told you, electronic microscopes are available, and beyond that so many techniques are available, we can have just a glimpse of it, and these all things are now adding up into the advent or the development of this biological science. If this all techniques will not have been there, then it may not have been possible that how it will be there. So it is a molecular genetics is there, it has been developed, that at what is happening at molecular level, at each step what is happening, what are the various chemicals, what are various proteins, what are various types of nucleotides, or whatsoever things are there, how they are doing, how they are participating into the activity, what happens because of one or other. So this was essential, and for identifying the molecular which are responsible for heredity. So the first one was, as I told you, it is Watson and Crick along with the Franklin and Wilkins. They figure out the structure of DNA long back, and then slowly one after another various mechanisms, various ways were developed by which the people, they could identify what is the reason, So earlier there was a restriction endonuclease which was isolated in E. coli, that opened the field of things that is called as a genetic engineering. That is, we'll go for this topic later. We call it as a recombinant technology. Or you might have seen that even bacteria causes what, they alter their DNA structure, and because of that they are able to go for resistance formation. Or similarly, we can insert some DNA portion from outside, and accordingly they will cause synthesis of something. So these are again a new way by which it is possible. Then these enzymes were used to linearize DNA for separation by electrophoresis and southern blotting. These are the techniques. Again, electrophoresis is one technique. Southern blotting is a part of electrophoresis, is type of electrophoresis only. Okay, but these are called as a southern blotting, northern blotting, or western blotting, depending on the either it is DNA, RNA, or protein. So it was able to identify electrophoresis technique itself having these names, that is called as a southern or southern blotting, northern blotting, or western blotting, and that gives idea about various kind of things are there. It is simple. You might have studied earlier somewhere electrophoresis also. I hope you might have somewhere studied, or you might have studied chromatography like thing, where you know that with the fluid whatsoever there, along with that the molecules, they are traveling, and because the changes in the structure, they will stop at one or other layer. Similarly here it will be, they will flow through the there, and there will be media will be there. On that media the electric current will be applied, charge will be applied positive or negatively, and on that basis the molecules will be attracted. A very small, very minute charge is there, and that molecules will be attracted toward the charge, and accordingly their movement on that particularly area, that particularly media will be different, and according on that basis of that it can be, that much portion can be separated out, and the area can be analyzed. So this is how this what we can say electrophoresis and blotting techniques, they are useful. Then next are the recombinant DNA molecules. That is, as I told you, it is a technology. We shall be seeing it at again. Recombinant DNA molecules means the DNA has been added with something artificially new, few more sequences are added into that, and what happens to that. And first recombinant DNA plasmid, you know about plasmid, you might have studied it in microbiology. You may be remembering, if not, please go through it, that these plasmids are responsible for having some extra genetic material or extra genetic information is there. Then later first DNA organism was developed by inserting recombinant DNA plasmid into E. coli, and that was called as a bacterial transformation. Then there was a development of DNA sequencing technique in 70s, and then this is a Nobel laureate, that is Frederick Sanger, who did a very important genetic research, and he enabled scientists to begin conducting genetic screens to relate genotypic sequence and phenotypic. Then very important, if you are going for or those people are reading about the COVID regularly, they must be knowing that there are techniques that is called as a PCR technique, that is what called as a polymerase chain reaction techniques. So this was invented in Mullis by 1985. It enables scientists to create millions of the copies of a DNA, same DNA, because artificially it will, or it's sort of artificial gene expression cycle. Okay, so one DNA will be copied into many DNAs, and that could be used for transformation or manipulated using the agarose gel separation. Later whole genome was sequenced, and then there was the eventual sequence of human genome. While the human genome project, it was a very big project actually carried out in 2001. Those who are interested can go through, they can read this human genome project, that almost how many genes are there, what genes are coding, what is the importance of that, what kind of various proteins can be formed, and it was really a very big study it was there. And because of that, a new field that is called as a genomics that has been developed. This is now called as a genomics, where the molecular structure of gene is related to the protein or the RNA, and then its functional expression, what happens to that. So this genomics is nowadays, or it is now growing like anything, because everybody understands one very simple thing now. Earlier we were going for, there are some signs, symptoms, and then trying something that this drug has given, and then this has happened. Nowadays it's different, that everybody now knows that there is a human genome project was there, and these genes are responsible for certain kind of structures or structural functional biological molecules like proteins, lipids, carbohydrate, etc. Then what kind happens because of that, what happens if these proteins are wrong, that means something is wrong with the genetics or in the process. And thus the studies are going, or they are directed toward that, what are the basic cause of the, even people were happy earlier to understand the observable cause, that there is called as atherosclerosis. If there is a cardiovascular problem, then somebody will find that there is a thing that is called as atherosclerosis. Why the atherosclerosis is there? Then because of the more lipid is there, and because of that there is a formation of atherosclerotic plaque, and so mechanisms are starting. Now people are going beyond that why few people are more prone for atherosclerosis, why few are not, because there are many relations are there. There are people who are having hyperlipidemia for long periods, still they don't have atherosclerosis. In other cases, even there was no big problem in the lipid metabolism, still they can have atherosclerosis. So people are going towards its genetic basis now, that although it was a study, this study shows that those who are having higher lipid or hyperlipidemia, they are more prone, but not all are prone. That means it is not only the cause, that may be something different cause may be there. So people are trying to work on that. Okay, and this is what important information, that is what importance in this. Then nextly, this genomics is, as I told you, it is linking the structure of gene to the protein, RNA, etc., and functional expression. There are many databases are available by National Council of Biological Sciences. Then there is Ensemble. It has been now merged. This genomics is simultaneously been hybridized or mixed with the computer analysis and computerized models, we call it as a. So these models and comparison of gene within and between different species is called as a bioinformatics, and the links of genetic mutation, what happens to that, then bioinformatics will able to predict the things. So this is a computer based model, that this is the gene structure and this should be the outcome. If something goes wrong, this should be outcome, and this is what there. So these are what various techniques are there. Basically they are divided into two important one, that is forward techniques. They will see what happens in the genetic mutation or genetic activity, how it will be there. And there are variety of such tests are there, where the genes are expressed, and because of expression, what are the outcome. So it is seen what happens if there is a mutation, what will be happen, if there is a transformation, what will be happen, whether there can be formation of resistance or not, if there can be receptor will be there, and because of the mutation, what will happen to the receptor, like that. So all these kind of studies are performed. This is done in a course of in the way of forward genetics, and it is the another one is the reverse genetics, used to determine the phenotype resulting from the intentional mutation in the gene of interest. So the phenotype is used to check, deduce here means to know, to understand, to understand. the function of the unmutated version of the gene, and mutation may be random or intentional changes to the genes can be there. So there are variety of mutations. You may please go through again mutation when you might have read in microbiology, missense mutation, and then one point mutation or point mutation, etc. So there are many such things are there. So that will help you to understand. Then addition deletion type of mutation. There are many type of mutations are there, the frame shift mutations, etc. So there are certain genes can be removed, and it has been tried very popularly for animals, particularly in experimental animals. So that particular important gene is removed. Example, if any gene is important for metabolism of lipids, then that particular gene will be removed, and because of that these animals will become hyperlipidemic. Okay, just like persons who are becoming hyperlipidemic. Then there are genes which can cause this maintaining the body weight. If this gene is removed, then the animals they will gain weight, they will become obese. So there are variety of such things are possible, and these things they are called as a gene knockouts. Knockout, just like it is used in boxing and other terms. So this gene knockout means that gene is removed, gene is not present, and this gene is, at least it is the taken care is that it will not express, means that gene will not be functional, that gene will not do any there. Okay, and these animals are used as models for various kind of such diseases. May it be hypertension, may it be diabetes, may it be other things. That what happens if these genes are not there, what happens to the physiology, what happens this. Okay, then some genes may be substituted into organism's genome, they are called as a transgene. Okay, to create to gene knock-in, means the gene which were not normally present in that particular animal or species, these genes are introduced, they are added into that. Okay, and they are called as a transgenetic or animals or transgene. Okay, so these transgenes are the gene is, or in opposite term, in earlier we have seen that some gene portion is not allowed to express, it is silenced. Okay, so we call it as a gene knockout. Here we call it a gene knock-in. That means a new this gene which was not there, a new activity, new function will be shown by that particular species or cell. So there are, these techniques have inherent bias regarding the decision to link phenotype to particular function. It is much faster in terms of production than forward genetics, because the gene of the entrance is already known. In earlier case, it was not known and it was just done. So this is how genetics is working. We can go for further parts into that with the nucleosomes and all. So this is how the genetics is working. Where various techniques are there. There are many techniques are there actually. That is, we can call it as a physico-chemical or biotechnological tools. And these all tools are helping. These all tools are adding into science by varieties of way, and therefore we are able to understand so many things. Let me tell you that this much name of proteins and all this was not at all known to anybody who learned the science during our time, because these are all in the very nascent way. Okay. However, nowadays it has been increasing like anything, everything is working, and this is how this science is working. Okay, so I hope you are understanding this. Please read this and read it. Whatsoever is important, make it. Whatsoever I have taught you, you try to make the notes. That will help you because it is a very vast science. Each of these can have its own book.